Right. Good evening, everyone. I'm Janelle Gurley, Director of Science and Programs at Mariah Mitchell Association. Of course, everyone on Zoom can't see me yet. And it is such an honor to host you all this evening for our complimentary Science Speaker series, which is generously sponsored by Bank of America, our lead sponsor, as well as Cisco Brewers of Nantucket and the White Elephant Hotels and Resorts. And it is such an earnest pleasure to present Dr. Val Hall this evening, as well as her Scallop research students who have been working tirelessly all summer to conduct research and have a full mentorship experience in the Scallop Lab here at Mariah Mitchell Association, which is also made possible by the Nantucket Shellfish Association, as well as the Great Harbor Yacht Club. And I will give a quick, brief overview and bio for Dr. Hall in the presentation this evening, and then I will turn it over to you. So this feature presentation will give an overview of the biology of the bay scallop and its unique fishery. Dr. Valerie Hall, Mariah Mitchell Research Associate, and 9 of her students, Ellie, Kinsella, and Grace Simanson, and if I said that incorrectly, please let me know. Sophia, Gus, Sayam, James, Kate, Julia, and Aiden will first describe the biology of the Nantucket Bay Scallop.
Oh, and Ilion.
And Ilion, did I forget someone? If I
Yeah.
forgot you, let me know. Thank you.
You didn't get on that list.
This presentation will cover the Nantucket Bay Scallop classification, its habitat, the distribution of the ecological niche, life cycle, reproduction, timing of the spawning, predators, parasites, and environmental hazards that the species currently face. Students will then describe the laboratory and field program, the mentorship component of this program, which is now in its 8th year here at the Mariah Mitchell Association. Without further ado and any delay, I turn it over to Dr. Hall. Welcome.
Right, so. We're gonna do musical chairs here. Yeah, okay. Oh, that works out pretty well. All right. I have to share my screen. This is about, I'm new to presenting this. I say yes. All right, then I go. Oh, this one?
Yep, and hit share.
All right,
2 more steps.
2 more steps. All right, okay, and then I can do slide. Okay, from the beginning. No, I think I forgot slideshow.
No, you got it. It's in it already.
Oh, it's in it. I just not used to all that stuff.
That's
at the bottom, yeah, oh, here we go. Oops. Okay, there we go. All right. So as Janelle said, 8 years ago, the program started, and Mariah Mitchell Association has given us a home every year. First in the Science Museum, the Natural History Museum, which is right across from here, for the first 6 years, and then we moved down to 33 Washington, across from the aquarium, and where we occupy 2 rooms instead of 1. So we expanded, which is great because we've also expanded in numbers. The first year was in 2016, funded by Shellfish Association. We had only 4 high school kids going into 9th grade from Cyrus Pierce School and 1 research assistant, and then all the way till now, where we have 20 students, 5 of them volunteers and 5 research assistants. And so one of the things I'm really pleased that in the audience is a former research assistant who was with me for many years, Mackenzie Welch, and we had to go through COVID together and everything. Somehow we kept the program going even then, even though it was just out of my house, but we've just had a great time. Every single student in the past 8 years has been wonderful. I've just really enjoyed working with them so much. So my funders, as Janelle said, the Shellfish Association for several years, Great Harbor Yacht Club for the past 3 years. I've also had other funding from Nantucket Marine and Nantucket Biodiversity Initiative, and several parents have helped out, which has been really, really great. So I'd like to start out by showing you who will be presenting tonight. Our volunteers, Ellie and Gray at the top. And then around the side, we have Sayam, Sophia, Gus, James, if he's on Zoom and we're still waiting for him to come in, but he's coming. Ilion, Julia, Kate, and Aiden, a great bunch of people. And that's half of the group. We had the first half earlier in the summer at the Great Harbor Yacht Club. So the bay scallop, scientific name Argopecten irradians. That animal's always fascinated me ever since I was an undergraduate and went to Woods Hole and had to do some experiments with predator-prey relationships. And I said, what an incredible creature. So I've tried to get that interest and excitement in the students, and I think they have learned too how exciting this animal really is. So I'm gonna start with some information about the bay scallop as an introduction. Aiden Haase and Ellie Kinsella will start in this introduction. And first, Aiden will tell you about the scallop habitat and range, and he comes from Maplewood, New Jersey, and he's a senior at the Pennington School, and this is his first year as a volunteer on the program. So Aiden.
So the natural habitat of bay scallops are shallow protected bays and coastal areas, which are both commonly associated with eelgrass, also known as seagrass, and sandy sediments. And then there are 4 subspecies of bay scallops. The first is the northern bay scallop, which you can see is found from the Massachusetts to the top tip of North Carolina. The second are the southern bay scallops, which are found mainly off the coast of North Carolina. The third are the calico scallops, which are found off the eastern coast of Florida in the Atlantic Ocean. And the last one, Val calls the Tex-Mex scallop, which is mainly found in the Gulf of Mexico.
And now Ellie will give the scallop's classification and describe its relatives and its importance to Nantucket. Ellie's a graduate of Nantucket High School and will be entering Northeastern University this fall to study chemistry and engineering. This is her second year in the program, this year as a research assistant. Ellie.
Okay, so scallops belong to the phylum Mollusca, and a phylum is a group of animals that share one or more fundamental characteristic that sets them apart from other animals. Other animals in the phylum Mollusca are cuttlefish, squid, land slugs, and gardener snails. Scallops are also bivalves, which means they have 2 hinge shells that protect their soft body on the inside. Other bivalves are oysters, mussels, and the New Zealand scallop. And bay scallops are very important to ACK. Nantucket has one of the last remaining wild bay scallop populations in the entire world. That's because they are in danger of extinction in their other ranges. Nantucket also has a large recreational and commercial fishery, meaning that lots of year-round islanders make their money off of bay scallops and selling them. And their fishing and scalloping is also a recreational activity that lots of families and friends enjoy that brings them together. Scallops are also an indicator of a healthy environment, like the old saying, canary in the coal mine, which basically means an early indicator of danger. When our bay scallops are dying or they're sick or they're not reproducing as they should be, we know that there's something wrong with the environment that they're in and that we need to do something about it. They also play an essential role in the coastal marine food web.
Thank you, Ellie. So we're going to go on now to the reproduction and life cycle of the bay scallop. Ilion Qureshi and Julia Winsor are now going to tell you about that. And first, Ilion will come up and describe to you the lifespan and the complex life cycle of this bay scallop, which is a very short-lived species. Ilion lives in Lincoln, Mass., and is a junior at the Moses Brown School in Providence, Rhode Island, and brought some friends with him as well. He's a longtime volunteer. He really has the record of the longest time in the program, which is 7 or maybe even 8. I've lost track. He started when he was 11, so and he's been great. So, Ilion, the floor is yours.
So starting off with the birth of the scallops, it is in what we call a spawn.
It occurs when the water temperature reaches above 18 degrees Celsius, and the adult scallops release eggs and sperm into the water that then come up. This sperm will when it is born, it is called a trochophore larvae. These larvae mature into a D shape that villagers and have their first shells for swimming. Eventually, they grow a foot that during the pedivelage stage, it helps them move around the water and settle down. When they grow older into juvenile scallops, the scallops attach to eelgrass using sticky threads, evading seafloor predators such as crabs and other things. Gradually, they become mature scallops, moving on to the seafloor. Once they reach adulthood, the scallops will spawn once in their only 2-year lifetime. Once they spawn, they are allowed to be harvested in the fall and winter seasons.
Now, Julia will continue this area by describing the unique way in which scallops reproduce and how the spawning periods can often give rise to two different kinds of scallops called on Nantucket classics and nubs. I don't know if anyone knows what they are anywhere else but Nantucket, but we know. Julia lives in Tarboro, North Carolina, is entering her senior year at St. George's School in Middletown, Rhode Island. And this is her first year in the program. Julia?
The bay scallop is a simultaneous functional hermaphrodite, which means that adults can release sperm and eggs in the same spawning event. The wider part of the gonad is the male testis, and below it is the female ovary, which can be either red, orange, or pink when ripe. Two significant spawns take place from May to June and mid-July, then again around August, September, and October. This first spawn are called the classics, as they have the larger growth ring around their shell. And the second spawn are referred to as the nubs due to their growth ring being smaller and towards the bottom of their shell.
Thank you, Julia. Okay, now we're going to go on to predators and parasites, and Sayam Thapamagar and Gus Simanson will now name and describe these major predators and parasites. First, Sayam will talk about the predators of both young and adult scallops and how the scallop protects itself from them. Sayam lives here on Nantucket and is a graduate of the new school. He'll be entering his sophomore year in Nantucket High School, and this is his first year in the program. Sayam.
Scallops live on eelgrass, and it allows them to be protected by predators since they're elevated on like the crabs and other organisms that eat them or on the beach floor. This is an example of one of the predators, a mud crab, which eats the scallops. And these are a couple examples of predators that eat adult scallops. We got the whelks, blue crabs, sea stars, gulls, green crabs, eider ducks, cornrows, rays. Each of them has a different method in opening and trying to eat the scallops. The whelks, they use their secretion in softening the shells of scallops, then uses its radula, which is a tongue-like organ with some sharp teeth to drill into the scallop's shell. And the crabs just keep picking on the scallop shell until they can fit their claw into it and eat the scallop. The sea stars uses its legs and muscles to pry open the scallop until its stomach is able to go into it and dissolve the meat. Then eider ducks just swallow the thing whole, and gulls drop them from a high elevation to crack open it and just eat the meat, while cow-nosed rays, which are mostly in North Carolina, they use their teeth to crack open the shells and then just swallow it. Then these are some ways the bay scallops can protect themselves.
they use their sensory eyes and tentacles to detect predators. And their hard shell, which is also used in camouflage, allows them to be hidden from predators. And the hard shell, since it's hard, it's hard to get into the their soft bodies. Then they have it, and unlike other bivalves, they have a swimming ability in the water, and they use their abductor muscles to close, and it allows for jet propulsion.
All right, nice stuff. All right,
At home, we can't see the screen.
Okay.
So we're going
All
to
right.
fix that.
make
so-
sure we're still seeing it.
Is this where we are?
Yeah. Okay, now Gray is going to come up and talk about the major parasite of bay salts, because they really don't have very many other, with special emphasis then on this pea crab that you see here. Gray is from Great Falls, Virginia, and is a senior at Tabor Academy here in Massachusetts. This is Gray's third year in the program. This year, she's serving as a research assistant. Gray.
So something we commonly find in our dissections with scallops are pea crabs, or oyster crabs, which are soft body crabs that are commonly found in bivalves. So they're known as kleptoparasites and steal food from the host to survive. And they generally find a host when they're both still on the smaller side. The bee crabs will remain free swimming and swim inside of the host. The males will be able to then leave the host later, but the females will grow bigger and then won't be able to exit. So they'll be stuck inside the scallop for the rest of their lives. And they latch on near the gonad, which unfortunately has a negative effect on the scallops themselves because the species actively feeds and it takes away nutrients from the gonad, which can mess with the fertility of the scallop and its reproduction. So the presence of pea crabs does have a significant effect on not just the gonad index, but also the body itself. In fact, its presence correlates with almost a 15% reduction in the mass of the gonad. And the prevalence of these pea crabs has also been seen to increase as the temperature rises, which can be a problem as ocean temperature rise for the scallops on Nantucket and other locations.
Great. So now Gus Simanson and Sophia Beni will highlight the major environmental issues that may cause risk to bay scallop. First, Gus lives in Great Falls, Virginia, and will be entering his junior year at the Thomas Jefferson High School for Science and Technology. This is his first year as a volunteer in the program, and he'll give you an overview of the environmental risks and discuss the importance of eelgrass to the scallop.
So first, we're going to talk about a couple environmental risks, and the first one is the loss of eelgrass habitat, which I'll talk about the importance of in a second. But basically, dredging and boat moorings, which disrupt the scallop beds, undermine the very foundation of the scallop population. And we can actively see this happening in Nantucket Harbor with all of the channel and ferries going through it and everything. And then next, harmful algae blooms. They pose a substantial threat to the survival of scallops because a study conducted by Woods Hole Oceanographic Institution found that these blooms release toxins that can harm the juvenile scallops and other marine life, which can create a very bad thing for our habitat. And next, also invasive species such as green crabs, because they burrow under the eelgrass beds, and that kind of disrupts the whole population. And pollution also, that has a damaging effect to eelgrass where scallops live. And climate change also disrupts eelgrass and the reproduction times of scallops. And then to the next slide, eelgrass is a crucial nursery ground for juvenile scallops because it offers protection from predators and ensures optimal access to nutrients. Because when young scallops, when they're first spawned, they go to the top of the eelgrass so they can get all the nutrients, and they're away from
like crabs and all of that. And then as they grow bigger, they slowly move down the eelgrass until they're big enough to be free. So more eelgrass means higher survival rate for the scallops.
Thank you. And Sophia lives here in Nantucket and attends Middlesex School in Concord, Mass. Where she'll be entering her junior year. And this is her first year in the program. She'll discuss the conditions which cause harmful algal blooms and then describe the one that we have most often here on Nantucket called rust tide. Might have to use the little arrow. Oh, you got it.
Okay.
Go ahead.
Okay, so harmful algal blooms, or HABs, are the rapid growth of algae or cyanobacteria that can cause harm to people, animals, or the local ecology. HABs can harm organisms by emitting natural algae-produced toxins, as well as depleting oxygen, often creating what are known as dead zones, and by generally clouding and overpopulating the water. HAB populations increase due to a number of reasons, both environmental and man-made. Toxin production can be stimulated by environmental factors such as light, temperature, salinity, pH, and nutrient levels. And fertilizers or sewage waste brought by runoff are primary suppliers of excessive nutrients. So around 2010, an increase in runoff and groundwater inputs led to the emergence of the dinoflagellate Cocodinium polycracoides. The dinoflagellate has a reddish color that filled the water, resulting in the name of rust tides. The dinoflagellate has a one of the most distinct instances of rust tides on Nantucket span from August to early September of 2010, mainly in the harbor, when scallops, filter-feeding organisms, ingest the dinoflagellate in high concentrations. Scallop seeds, as well as some larger scallops, are killed. Even if the scallop isn't killed, the dinoflagellate can decrease the body mass substantially. Rust tides, as well as other harmful algal blooms, are a major factor in the population decrease of Nantucket space scallops. And this is what the dinoflagellate looks like.
Nice job on the pronunciation of that thesis too. That's not easy to do. I just want to give a little overview of the field and lab program. It's a 12-week program. Basically, the students come for at least 5 weeks during that time and go 2 or 3 days a week, and they have to earn a minimum of 30 volunteer hours, and they get credit for that. Most their schools will accept that as volunteer credit, community service. However, that's just a minimum. I've got some people that are up in the 40s and 50s as far as how much they contribute. And so far, we've had 7 collecting trips every 2 weeks. Some of you may know Carl Scholand. He's just an incredible guy, and the kids really love him. And he takes us out in a scalloping boat. We bring back about 60 scallops every time. And during a 2-week period, which is what we hope will be, we will go all the way from dissecting the scallops to processing them, embedding them, sectioning them to very thinly, staining, and finally looking at the prepared slides. We've had a little issue with some of our equipment dying on us this year because it is old. So we're not quite doing it every 2 weeks, but we're getting as much as we can done. They keep a daily log set up just the way a scientist would do it. And they also participate in a group presentation like this one here. And there was an earlier one at Great Harbor Yacht Club, and periodic lab seminars where we just talk about what we're doing in the lab. And not to bore you with this other next slide, because these are all the steps, but I'm going to have the kids explain each step with lots of pictures and everything. So we can pretty much just look that over. So we'll go on to the next one. Let's see what I want. Okay. Kate Kinsella will now discuss collecting trips with our boat captain, Carl, and what we do with the scallops when we get them back to the lab. Kate is a Nantucketer in her first year as a volunteer in the program. She graduated from the new school and will be entering her freshman year at Nantucket High School. So, Kate.
Okay, so
Usually per year, we go on 7 trips that take from 2 to 2.5 hours. We go to 6 stations on each trip, and some of those stations are East and West Hashi Shoal and First and Second Bend. Usually we go with 4 volunteers and 1 research assistant who leads the collecting trip. Once we get to a station, we get the surface water temperature and the seafloor temperature. Then Captain Carl drags a dredge across the seafloor. Usually, we get about 10 scallops that we can dissect after, and sometimes we also get some sediment off the seafloor too. So then we sort through what comes up from the dredge, and we get 10 scallops and classify them by their nubs or the classics and how old they are. And then we make a note of everything in a notebook.
Change to the pictures. Yeah. There they all are having a lot of fun.
And then back at the lab, we usually get 60 scallops that we can dissect, and we usually dissect 12 each. The first thing we do is we get the height, width, and length of each scallop. We open the scallop and take the body out. Sometimes, as Gray described, we find parasites in the scallop. Then we weigh the body of the scallop in grams. And then we find out how ripe the gonad is by the color scale. The color scale is from 1 to 5, and the more orangey red, the more ripe the scallop gonad is. And that means the higher number it is. Then we separate the gonad from the body and we weigh the gonad. And then we sew a label with the date of the collection and the ID number of the scallop. After that, we put the gonad in 10% formalin to preserve it before we put them into cassettes for embedding.
Thank you, Kate. Has our next person, has James, okay, well, James, the person who was going to come on Zoom may have had some issues with getting on, but he was just going to describe what we do with the data. So you see some people entering data, and it's all raw data, and from the raw data, they put it on an Excel sheet. And this is just one little page of it, and all the things that Kate described, shell height, length, width, where the growth rings are. These are all 1-year-old. This C1 is first year classics. They only live 2 years, so we don't find very many 2-year-olds. They're 10, 15% of the population compared to the 1-year-olds. With the color number, as Kate described, and then the weights from the gonad and body weights, we can get a GSI, gonadosomatic index, which tells you how ripe the scallop is. And then we always put down the presence of pea crabs or any other things that we might see that are kind of unusual shell deformities, things of that sort. So next, Gray, the research assistant that you already met, will now go through the steps of how we prepare the specimens after they're dissected and put in formalin. And eventually they get embedded in paraffin. And thus, all of these things are going to get them ready to do the thin sectioning to make slides. So Gray.
So post-dissection, we have to prepare the specimens for the microscope, and the first step of this process involves getting them into plastic boxes called cassettes, which are the little white things in those jars right there. So this is done by taking a razor blade and cutting off a small portion of the end of the gonad, making sure you get both the testes and the ovaries. So you can see that when you're on the microscope. Once we do this cassette, once we take the section, we place it into the cassette and then place them into various dehydrating solutions. So here we have alcohol in order to prepare it for the embedding process. So on the left, we have an image of the embedder. So the embedder basically works by heating up paraffin to a high degree, and you use that and pour it into where you have the specimen. So over on the bottom, you have the specimens in the cassettes, and then they're placed into metal boats, which basically act as a mold for the paraffin to set and make sure it can fit into the embedder. So there she's placing into that, and then you have to trim the blocks down because the wax kind of gets everywhere. And then it finally creates these blocks that can then fit into the microtome later, which you can use to create slides of sectioning. One more. So here we have the microtome, which is everyone's favorite part to do. So basically you stick those blocks into the microtome and then you crank it, which forces the razor blade to come forward and take a section off the block. And the goal is to get a really thin section that it's easier to see under the microscope later, but still make sure that it's more put together than like broken up. You have to be really careful in order to actually have a full specimen that can be looked at later. But yeah, here's a microtome and here's the blocks that you would create in the embedding process. So that's
Thank you for. And that microtome, one of ours broke this, that's the actual one that died, the one you saw in the last frame. And we're very sorry to lose it. They don't make them anymore. It was about 50 years old, so, and it was bought with Shellfish Association money way back. And we're very, we should conduct a funeral or something for it, but we're really missing that one. So lastly, Ellie, who's also met before, one of our research assistants, will continue the process to the final product. First, she'll say anything else she wants to say about sectioning, and then staining the resulting paraffin ribbons that we get from sectioning, and then explain how we evaluate the slides under the microscope to assess their spawning status, which is the whole point of going through all of the stages. Okay, Ellie.
Okay, so it's kind of hard to see, but this little black rectangle right here is called a slide warmer. And when we get sections off of the microtome, we get 2 ribbons, is what the sections are called, per cassette. And we put them on the slide warmer and we leave them overnight so that they can dry. Then in the morning, we label them and we look at them and see which ribbon and side has more of the whole gonad on it. And we take the better side and put it in a staining box. And then the staining box can hold about 12 slides in it. So we tried to fill up a staining box before we do a staining. And then we move on to staining. It takes about 30 minutes overall to do a staining because there are multiple dyes and chemicals that it has to go through. The slides go through alcohol and slide bright, as well as eosin, Scott's bluing, and hematoxylin, which all try to brighten up the ovary and testis so that we can see it better under the microscope. So after it's gone through the whole staining process, we then mount the slides, which means that we put mounting medium on it and a cover slip on top so that we will be able to look at them under the microscope. And then we move on to microscopy. So we use Sastry stages to identify what stages of spawning the gonad is in. So a gonad is both an ovary and a testis, and the ovary and the testis might be in different stages of spawning. So the ovary might be in stage 2 while the testis is in stage 5. It really depends. And we make very detailed notes in our lab notebooks of what stage they are in and what we see under the microscope. And this is an image of what we might see under the microscope. We look for many things, but mainly we look for mature oocytes and immature oocytes. The mature ones grouped together in what's called a follicle. And we also look for something called atresia, which is over there in the left. It kind of looks like the oocytes are swirly. Atresia is caused by many things, many changes in the ice scallop's environment, like higher or lower water temperatures, not enough nutrients, or the presence of some kind of stress from predators.
Thank you. Okay, to wrap things up, we have some outcomes of the research project. I mean, one that isn't on there, I think we have fun. And that science should be fun. It should be exciting. But we're trying to examine the effect of climate change on bay scallop reproduction. And I don't know why this part of this is off, but it is. Anyway, one student was from 1 year, a couple of years ago, was looking at the average of the 2 surface and bottom temperatures, and these are Fahrenheit over the season, and you can't see the dates all the way, but they go from early June all the way to late August, and you can see that by late August the temperature was 75.2 Fahrenheit, and however this year the temperatures those kinds of temperatures were found in mid-July, not the end of August. So something's going on. So we're trying to correlate that with what's going on with the spawning status of the bay scallop. Another big outcome and important is that the kids get to experience working in a real research laboratory. It doesn't have modern equipment. It all came from eBay or wherever we could find it, beg, borrow, whatever. But it still works and they learn the principles. Mastery of scallop biology, I think you can see they've learned a lot about scallops and their ecology, reproduction, etc. They mentor each other. So we have research assistants mentoring volunteers mentoring each other. It's just a really good environment. Everybody shares information and helps one another. And also collaboration and telling each other what they've learned. And fostering stewardship of Nantucket waters. I think they already have whether they live here year round or whether students are only here in the summers, but they've been coming since they were babies and they do care about the waters around Nantucket. And this just makes them maybe have a better idea what might be done to help. So I'd like to end by just thanking my funding sources. And I wish Karen, maybe she's on Zoom, but Karen Ketter has been my liaison to the Great Harbor Yacht Club Foundation and the Mariah Mitchell boards. And I also call her my guardian angel. So I hope she hears my thank you. And Captain Carl is our main person who helps us, but we also had Bruce Cowan help as a substitute when Carl wasn't able to do it. I've told you we also had some other funding. This is a whole group from the entire summer, 5 research assistants and 15 of the volunteers. So your child's name is up there, but there's also other kids that did their talk earlier in the summer. So thank you all for coming. If you have any questions, please feel free to ask. I'm going to see if maybe the students will try to answer them first, because I think they should know the answers. Thank you. Any questions?
What questions do you have? Certainly.
Did they answer all your questions? Oh, Mackenzie.
So I guess any student feel free to answer this, but what has been your favorite part of working in Val's lab?
Repeat the question for those at home.
I should forget. Yeah, the question is, what is your favorite part of working in Val's lab? Anyone? Ellie.
I mean, one of my favorite things, especially over the past 7 years, was just kind of growing up throughout the summer in Val's lab, and she teaching me a lot about how the ocean works and how marine biology works. Taking a big interest, everything like that. I've done a lot of volunteer hours.
Anyone else?
I think you can all share what your favorite part of the experience has been.
Would you like first?
I'll go.
All right.
Okay, good.
Sophia?
That's fine. You're gonna have to get
So I really enjoy that it's like interactive classroom experience, but without the stress of grades and things like that. So you get to really focus on the things you're interested in, and it's like helps me kind of rediscover my interest in marine biology, because I have like that for my whole life, but it's difficult to just focus on the interest of it in the classroom and worry about other things too. So I think this is a good opportunity.
Thank you, Sophie. Oh, another question. Yes.
Does anyone not like to eat scallops now?
Oh, the question is, does anyone not like to eat scallops now?
I love to eat scallops.
It's interesting. The days we actually dissect, I don't think they seem very appetizing when you're actually dissecting them, but how many of you still eat scallops? What do you- But they're out of season now, so they're not really very tasty compared to how they would be in the winter. That's true, but we do have a permit, so And we don't take very many, so we're allowed to, but we don't eat them. But, yes, sir.
I have a question for some of the students. Do you have, knowledge that you get when you're reading about scallops? I've always heard for the two eyes, that they're really reflectors. And if you have a conversation about that, I found recently was Martin, so the PhD about that's out of, Ed Wong's West Whale. And I found out something about, the fact that they're not like us, who have eyes, they put our brain, we put an image together, they use theirs more along this touch. And then I discovered that touch is from the ball. It's kind of great, but it's a great book to read, guys. When you're in science or anything, that's not.
So, yeah, so the eyes of scallops, the question is, do they just have 32 or can they have a lot more? And are their eyes more than what we used to think? I mean, are they more like human eyes than we ever thought? I think I've read that same article. It was really amazing. Have any of you done any research into the eyes of scallops?
No, not particularly.
No. I know one summer, the students tried to preserve the eyes and then section them, but they're so fragile, it was hard, so we could look into the eye and see what they look like. But I think it would take different techniques. Yeah, right. Very. But the scallop senses in general are incredible. They have such good senses.
So one of the things that we forgot on something, right? That scallops down the ladder and to the base, to the mud, whatever, as good data. I have no idea. These things that we build grass.
Oh, yeah. That's a very, it's not that, it hasn't been known that long that they do that either, that the young scallops stay high on the eelgrass and then later, partly because they get so heavy, they can't hang on by that foot anymore. The foot is too weak, so they fall to the bottom. Yeah. But so that was good that you folks brought that up. Any other question? Any questions on?
I have a question.
Yeah.
Not on Zoom yet. At home, if you have any questions, feel free to type them into the Q&A box and we will answer them live for you. My question is, over the 8 years of all of the research that you've conducted, where are you at in terms of the correlation with the spawning and the temperature rise?
Yeah, well, it's still a work in progress, but it's definitely, it's difficult to figure out exactly what it's doing to the spawning cycle, because every summer is different, has so many different conditions. But in general, it looks like the early spawn is getting earlier and the late spawn is getting later. And then there's more of a in-between time when they can't spawn it on because it's too hot. So temperature is definitely involved, but we don't know if that's a bad thing for the scallops or not. I was talking to Tara Riley at the hatchery, and I said, if they're spawning at the wrong time, or is there larvae, are there larvae that are starving because there's not enough plankton? And she says, well, there's plenty of plankton out there, so apparently that's not an issue. It is with some fisheries, but it doesn't seem to be. So it's very complex.
And do we feel like we're seeing any rebounds in the scallop
Yeah.
size?
This summer, I hear that it was in the front page of the Inky, I think last week, that there are tons of scallops out in the harbor now. And if they survive, we should have a couple of really good seasons. But because they only live 2 years, it's going to be fluctuating. And if in the 80s we were getting 100,000 bushels in a season, and now we're getting 3,000 bushels, even if it goes up for a couple of years, is it still going to have the downward trend? Because we have a problem with the environment, as the kids were saying, what's happening in the harbor and the canary and the coal mine effect, the sensitivity of the bay scallop. But what do you think, folks? Is this animal worth saving?
Yes.
Any reason why you think that's true? So what if the animal goes extinct? No comments? That's my question to you after the summer.
I feel they have like an important part in filtering out pollutants in the harbor, and without like them as playing that part, and every other like organism.
Yeah, and you lose one organism and you don't know what the effect's going to be in the rest of the food web. Or in other ways they affect each other. Yeah, so, it does affect, yeah, if one animal or plant goes extinct, it affects many others, yeah. Including humans. Yes, Laura?
Well, sorry for saying back to the meeting Winsor for all the kids. Been awesome over the years. So I guess an important question for me is, this is your last summer, and this is the last year of the program, which, you know, I had 4 boys go through the program, and Zara was 10. I was hoping she could eventually go through the program as well, and so selfishly, I'm like, what's gonna happen? Because I think it's such amazing information and knowledge, especially from Nantucket. Where do you foresee, what would you like, if you could wave a magic wand?
If I could wave,
What would that magic wand look, what would that, what would it look like?
well, thank you, Laura, it is my last summer of leading the project, and if I could wave a magic wand and find somebody who wants to take over, who's either crazy enough or whatever to do this, or loves scallops so much, and loves working with kids so much, that I would be really happy that this would go on. And if the funding sources want to continue this, it would be great. So that's my pitch, I guess. If there's someone out there that's dying for a really exciting and rewarding thing to do in the summer, let yourself be known.
Thank you for everything that I've sat through a number of years. It's amazing. You never know where the next year leads. But
Yeah,
I appreciate it.
thank you. Anything else from anybody? Any questions from any students or parents or?
This presentation was really good. It was really beefy for scalloping this year. There's a lot of information. Compared, like, I just feel like every year I've seen this presentation from you and the students, it just gets better and better.
Oh, that's good to
So
hear.
that's why
The presentations are getting better. And then students are just, this is a great group of
Yeah,
students
yeah.
this year, really. All of them, it's been really fun to be with, serious in learning and, just great group of people. So I'm gonna miss you guys. So.
On behalf, Val, on behalf of Environmental Association, we're so indebted for everything that you've done for students over the years, and not just for the Environmental Association, by weight of us and the Shellfish and Great Harbor Yacht Club, but as a former educator myself, I just personally like to thank you for your years of teaching service. It means a lot.
This has been a highlight of my career. I taught for 35 years, secondary level science, but having this program, working with students like you all, has been a highlight, a big highlight of my career. So, yeah.
Extremely grateful that you've imparting all your knowledge and the small fishery with us. But seriously, everyone, thank you so much for joining us this evening. It's been such a pleasure to host you all for our Science Speakers series, and without our sponsors and without wonderful budding researchers and scientists like yourselves, this would not be possible to present to everyone. So thank you for your weeks upon weeks of hard work and your enthusiasm, and Val, thank you for your mentorship, your stewardship, your leadership. And with that, I say thank you and have a great evening, guys. It's been a pleasure.
All right.